Hello again. Now we will continue with chapter 7. So chapter 7 and 8 are the most intense and potentially challenging content of this course. So yeah, please pay attention and make sure you try to understand everything. There's also a bit of math involved. It's not complicated math, just simple arithmetic mainly, but it will involve type of analysis that you might not be so much familiar with from other biology courses. I think it's definitely useful, and honestly, this type of simple modeling that we'll cover here is very, very simple, very basic, but it's good for you to become familiar with. In biology is becoming a more quantitative field, and these skills will be definitely useful, not just for those of you who might continue studying evolutionary biology, but also those of you who might study other aspects of genetics or ecology, um, yeah, in kind of field, or structural biology. Okay, so here we'll be studying simple models of population genetics of allele frequency change. We'll start with the Heidelberg equilibrium, and also go into simple selection models and mutation models, mutation selection balance. And then in the next chapter, we'll also start considering genetic drift. Okay, so actually this type of analyses, it goes back to early 20th century, when again people were discussing the validity of the Mendelian model, and there was this specific discussion of why Mendelian traits, for instance polydactyly, which if you have, oh sorry, this is not polydactyly, another another phenotype called brachydactyly, with somewhat disshaped fingers. Why we don't see it more often in the population? So the some scientists were arguing, okay, so if in the Mendelian model we expect one, so this is a dominant trait, as you see, and if we have two heterozygous parents having kids, 3/4 of the kids are expected to have to show the phenotype, and only 1/4 will not show brachydactyly. So why the question was then, why don't we see this pattern more often in the population? Why doesn't the population show 3/4 of brachydactyly? And what later these scientists realized was that they were confusing what you expect, so the patterns of frequency you expect to occur in a family in the Mendelian model, relative, and what you expect from the behavior in a gene pool. So this type of gene pool level of thinking wasn't around then, and what Hardy and Weinberg, these two British and German, I think, natural scientists at the time, they proposed was filled in that gap. Yeah, so early 20th century, people were discussing whether the Mendelian model would be correct, and why don't we expect, why don't we observe the frequencies we expect in the F2 ratio, in the F2s across the whole population, or why do recessive traits not get lost, etc. And yeah, so thanks to developments again about 100 years ago, people started thinking about genetics and biology at more quantitative levels, and they started trying to develop models that could predict how populations would change over time. with respect to allele frequencies and the phenotypes affected by those allele frequencies. So, for instance, through these models now we can address questions like how fast the new mutation will fix in a gene pool under drift or under selection. So we can predict on average how the rate of events of change. We can also predict conditions of equilibria. So when a population reaches a balance such that neutral mutations come in by mutation, and then, or I can say neutral variants come in by mutation, and then some of them are lost by drift. On average, through this incoming and outgoing process, how much polymorphism do you expect in a certain population? If the population size is X and mutation rate is Y, what do we expect? And we'll learn the answer to this in the next chapter. And you can also involve selection, and you might ask, okay, how much polymorphism do you expect under balance and selection? Okay, so when we say equilibria, we're talking about the state of a population with respect to genotype frequencies, and the population might be under, for instance, forces of mutation, drift, selection might be pushing a population in the genotype space towards different directions, and these push and pull effects might eventually lead to a state where the population has reached what we call a stable equilibrium, such that it does not move further, and any push that might move it around would be countered by opposite effects, so that it would move back to its original position. So this would be a stable equilibrium, just like you can imagine the population is a ball, and if it's in this type of structure, this position will be the stable equilibrium. Any force that moves it to a higher position will be countered by gravity, and it will fall back to this position. So this will be a stable equilibrium, and we'll learn how. We'll see examples of these. You can also have a population of an unstable equilibrium. So currently, it might reach a position where there is no, where it does not change, but it does not move. But any slight perturbation will lead to an accelerated rate of events, so that it will move away from this point. So this would be an unstable equilibrium. Without perturbation, it does not change, but with any perturbation, it will move somewhere else. You can also have a so-called neutral equilibrium. So there's no force pushing the population in any specific direction. There's also and at any position it can remain. So there's no force that makes it more likely for the population to be here than to be here. And you can also have a mixed equilibrium, such that you can have forces that move the population in one dimension. Here, let's call this the x-axis. Any push towards this side or the other side of the curve upward would be countered by gravity, and the population will move back to this lower position. But along this axis, let's call it the y-axis, along the y-axis, there is no force that makes the population more likely to be here than here. Okay, so these are different equilibria: stable, unstable, neutral, mixed, and we'll see examples of these. First, the first model we'll be discussing is the Hardy-Weinberg equilibrium. This is also the null model of evolution, basically describing gene pool where there's no evolutionary change at all, and it allows us to make predictions on if forces that drive evolution are not strong, uh, are not affecting allele frequencies strongly, what kind of expectations do we have? So it will be described by the Heidelberg equilibrium, and basically it allows us to predict given allele frequencies in the previous generation, call it T. It allows us to predict the genotype frequencies at generation T plus 1 under specific circumstances, which are basically no evolution. Yeah, and once it also predicts that once genotype frequencies are established, the population, again without while no evolutionary change is happening, the population will remain stable. Both the allele and genotype frequencies will remain the same. And what are those conditions? So we assume a gene pool where there's a population is diploid. There are discrete generations, they don't overlap. And there is, first of all, non-random mating. All individuals just contribute their gametes to infinite size gene pool, and then the next generation is created by randomly drawing 2 alleles per individual from this gene pool. There is no migration coming in, going out, no selection, so all alleles same. So we also assume biallelic polymorphism, so 2 alleles, and the alleles have equal fitness, and there's no drift, so the population has infinite size, and the allele frequencies of the previous generation remain the same in the next generation. That means no genetic drift. There's no mutation changing allele frequencies. Yeah, and among these factors, I might note already that random mating, the population being totally homogeneous, is the one which is most frequently not fully valid in natural populations. Usually in populations, you don't have individuals having access to every other individual for mating. But these other processes, of course, are also at play in natural populations at different levels, but not all of them at the same levels. Okay, so okay, this is the model. We have the parents. They create a gamete pool that reflects their allele frequencies. Everybody contributes their gametes so that the allele frequency here in the parents and the allele frequency in the gametes is the same. And then offspring are chosen from this pool by choosing 2 gametes randomly. That's representing random mating to create diploid individuals. Allele frequencies stay the same. This model allows us, the assumptions we discussed, allows us to calculate the, given the allele frequencies of the previous generation, we can calculate the genotype frequencies of the next generation. And that will be this formula that you already know from high school. So homozygous individuals A1 will occur at the frequency of A1 squared, and A1, A2 heterozygous will occur at the frequency of 2PQ. Why 2PQ? Because it's basically the combination, right? You can have heterozygous individuals when the first allele is A1 and second is A2, or vice versa, first A2 then A1. there are two alternative paths to the same event, then you sum up those probabilities, so you get 2pq, and this is q squared. And here we see how the frequency of heterozygotes changes depending on frequency of A1. And as you see, the highest number of heterozygotes you have in a population when the allele frequency is intermediate, when both A1 and A2 are at 50% frequency, you have highest frequency of heterozygotes. And yeah, so we basically, given the allele frequency of the previous generation, you can estimate the frequency of genotypes in the next generation. Please make sure you're okay with what allele frequency and genotype frequency, what these mean. Allele frequency is the frequency of the alleles in the gene pool, right here. Genotype frequency is the frequency of the diploid genotypes, of the 3 different genotypes. And okay, so this is this model describes a mixed equilibrium. Why is that? Because you can, for instance, start the population which is not in Heidi-Weinberg equilibrium. You could have a population which is composed 50% of A1 individuals, 50% A2 individuals, right? In this population, the allele frequency of both A1 and A2 will be 50%. And then based on this, given this situation, in the next generation, if this model works, if the assumptions of random mating, etc., hold, in the next generation, we'll have genotype frequencies that are described by the Heidi-Weinberg formula. Allele frequencies, in a way, won't change. And so you can predict the genotype frequencies. The genotype frequencies will change from one generation to another, and then they will remain the same thereafter. So with respect to genotype frequencies, here shown on this axis, with respect to genotype frequencies, Heidi-Weinberg model predicts change. It can happen if the population is composed only of homozygotes or only of heterozygotes. In the next generation, through random mating, we will have a change in genotype frequencies from here to here, for instance, or here to here. But the allele frequencies won't change. There's no force changing allele frequencies from one generation to another. That's why it's called a mixed equilibrium. Okay, yeah, so assuming genotype frequencies are not in equilibrium, in the next generation they will reach equilibrium, allele frequencies won't change. It would have been a stable equilibrium if there was another force. If there was another force affecting the allele frequencies and pushing them towards, let's say, this point or some other point, we could call it a stable equilibrium, for instance, by selection. Okay, and let's do this exercise. Myoglobin alleles in Japan. People measured 2 versions of myoglobin in the Japanese population given a sample. And in the sample, A1 heterozygotes had 59% frequency, A2 8%, etc. So now our question is this locus at Heidi-Weinberg equilibrium? If it is not, what this could mean, we will discuss a bit later, but first let's try to decide if it's in Heidi-Weinberg equilibrium. How do we go about this? We first need to guess what the allele frequencies will be. And to estimate the allele frequencies, you might think, okay, let's run the Heidi-Weinberg formula in the reverse way. So if these are the genotype frequencies, we could take the square root of these frequencies, and that those should give us the allele frequencies. But that's not the correct reasonable way of thinking, because that assumes the population is already in Heidi-Weinberg. But this is something we don't know. Perhaps it's not under Heidi-Weinberg equilibrium. So what we should do would be to collect to calculate allele frequencies using all the data. And how do we do that? For instance, the A1 frequency will be the frequency of A1 heterozygotes, and half the A1, A2, sorry, A1, A1 homozygotes and half the A1, A2 heterozygotes. And that gives us a frequency of 0.755. And we can also do the same for Q, or explicitly, or we can just simply subtract 1 from P. Yeah, that would give us Q. You can do this just to confirm your to make sure your calculation is correct. And yeah, so it is important. We cannot assume if our unless we're explicitly told the population is under Hardy-Weinberg equilibrium, we cannot just take the square root of any homozygote and to calculate the allele frequencies. We have to calculate them explicitly from the empirical data. And then given the allele frequencies, what we can do is to estimate the frequency of each genotype, each of the 3 genotypes. And if we do that using the Hardy-Weinberg formula, these are the numbers we get. So as you see, pretty similar. And then, but how can we decide if these frequencies actually are really different or not? Well, we need actually just the frequencies won't be enough, or the proportions won't be enough. We'll need the actual the number of observations for each of these 3 classes, and then we can do a chi-square test, for instance, right? Chi-square goodness of fit test, and one could also use Fisher. Well, chi-square actually makes more sense, and you would have 3 sets of observations, 3 categorical, 3 types of categories, each with the number of observations, and then we can ask, do these numbers fit what we would expect here? This type of calculation you'd already done in secondary biometry, you might remember. And if you run this, it turns out that there's no significant deviation. So the numbers don't significantly deviate from what we would expect from Hardy-Weinberg. What does that mean? Well, does that mean there's no evolution going on at this locus? This is something we'll touch upon. We can also briefly discuss, so assuming the difference was significant, perhaps with larger sample size, we might have found similar proportions, but the difference might be called significant if we had larger sample size. What do assuming the differences do mean something between the observed and expected, what is the deviation pointing out? When you look at these numbers, in what direction is the deviation? So which type is occurring, which type or types are occurring more than expected and less than expected? And you should easily notice that the heterozygotes are observed less than expected. The homozygotes are observed more than expected. And why could this be happening? Very simple. You might imagine different answers, but a very simple reason is non-random mating. Substructure in the population, so individuals of one type tend to be living, let's say, in one region of Japan, individuals of the other type, another region of Japan. So the frequency of homozygotes, this kind of situation will increase the frequency of homozygotes compared to the Hardy-Weinberg expectation. Of course, you could also have some degree of migration into the region which is being studied. Selection could also have a contribution, but kind of less likely. Most simple, straightforward explanation would be non-random mating. The other question, does this mean there's no evolution at this locus? Answer is no, it doesn't mean there's no evolution. It just means that we cannot detect evolutionary forces just within one generation at this locus with this sample size. Okay, so just whatever processes are happening. They're happening at a very subtle, low scale level, and with our given sample size in just one generation, we cannot detect it. But if we had been studying this locus over more higher number of generations using larger sample sizes, we should be able to identify effects. There might not be selection indeed, but there would be some amount of mutation happening and some amount of drift happening. It will vary, of course, across from population to population, locus to locus, but drift and mutation are always happening. Selection is sometimes happening, but these effects are weak and so might not be detectable in a single generation. Okay. So either by studying the population in the long term or using very large sample sizes, we can identify these. Okay, so now we go beyond Heidelberg to more serious models of evolutionary change, and we'll start with the selection model of directional selection. And you might remember this case, selection on mouse coat color. In this, normally mice are living in light desert environment in Arizona, Nevada, someplace like that. And then there's, due to lava flow in the past, there's some part of the desert where the soil is black. And then a black type has dark coat color type has colonized that area, and want to see what the selection, if we have originally white type, light color type in this area, and a dark coat color variant appears, what would be the selection on this variant? And given the selection, how fast would it spread through the population? Okay, so how much selection and how strong, given selection, how strong will allele frequencies change? Assuming selection is the only force. Okay, so the variants were already, variant here was already identified, variant in the MC1R gene. And the dark allele is dominant, it turns out. And it also turns out the mismatched light mice, so the ancestral, actually these are the ancestral mice, if you, they represent the ancestral phenotype. If you put them in a dark environment, their survival rates are 60 to 98% lower than the dark individuals. Okay, so they survive worse, which means dark individuals survive better. And now, given this, we'll discuss what kind of selection is happening, how much, and how fast selection will work. Okay, so the name of the selection process that we'll be studying is directional selection. It's selection towards one extreme phenotype, as you might remember. We discussed in the first chapter, directional selection. And it's a type of frequency-independent selection. That is, the allele frequency doesn't affect fitness. Fitness doesn't depend on how common or rare the allele is. We'll see other examples later where fitness depends on the frequency of the allele. So those are called frequency-dependent selection. This is a case of frequency-independent selection. And yeah, one phenotype allele is favored, alternatives are disfavored, and yeah, we can talk about positive selection on the favored, negative selection on disfavored alleles. Okay, and selection coefficient we can try to estimate given these values. So selection coefficient shown in S, you should remember this, both name and the symbol. S stands for selection coefficient, and it's the difference in fitness. Okay, it can be used to represent increase in fitness or decrease in fitness. Here, it's described as a reduction in fitness of a phenotype relative to alternative. So we can say the fitness of the dark phenotype, like dark coat color phenotype in dark environment. And of course, fitness always depends on the environment. So here we're only discussing fitness of these two alleles in the dark environment. In light environment, everything would be vice versa. In the dark environment, fitness of the dark phenotype is 1, and fitness of light phenotype is 1 minus S, okay? Lower fitness. S is a positive number, so this will be a lower fitness. And if S would have been 0, then there would be, if selection coefficient was 0, then the two fitnesses would be the same. The allele would be neutral, the phenotype would be neutral. And if S were 1, then fitness of the light phenotype would have been 0, so would be basically lethal or just not transmissible in this environment, like a dead end. But in this case, okay, what's the, we need to calculate S. And here is a table showing us the genotypes and the fitness effects given, and here you see that the A1 corresponds to dark, you can imagine is representing dark allele and it's dominant, so the heterozygotes also have fitness 1. Okay, although they carry only one copy of the dark allele, and only homozygous light color mice have fitness 1 minus S. Okay, so here what would be S? We already said the light colored mice in this environment have 60 to 98% lower survival than the dark colored mice, and ignoring reproduction effects and just focus on survival, we can estimate S here is 2% to 40%, and we can just use the average of these. Okay, so yeah, here we only take into account survival. Yeah, and also, but we assume that the allele's fitness is independent of its frequency in this model. Okay, so let's imagine to see how allele frequency change would happen. Imagine a population that starts with 50% A1, 50% A2 types, and imagine we have all three genotypes also at the equal frequency. So 100 A1 homozygous dark, 100 heterozygous, and 100 heterozygous light, homozygous light individuals. And okay, so we can imagine to make the scenario more realistic, all these groups, we introduce these mice, these 300 mice in the environment. Let's imagine 40% of all groups die first to random effects, but then there's also selection against light coat color and for dark coat color coming in, and the selection coefficient, let's choose 0.1, so some midway between these two. So assuming selection quotient is 10% against the light phenotype, so 90% less survival of the light compared to dark, we would have these numbers at the end of the day that would be able to, produce the next generation. And as you see, so these mice, only 60 dark left, 60 dark homozygous left, 60 dark heterozygous left, and 54 A2A2 light phenotype mice left. Let's imagine they mate randomly and create the next generation. But the numbers in the next generation, even if we imagine 300 mice are produced, the genotype frequencies will be different than these ones, right? Because as you see, the A2 alleles overall frequency has decreased, right? So here the A1 and A2 allele frequencies were 50%. Here it's so obviously lower, right? A2 is lower, A1 has become higher. And if we do the same experiment one more round, A2 will further decrease and further decrease, etc. A1 will be increasing at the same time. Okay, so and now our goal is to come up with a formula that will describe this change in allele frequency given S. Okay, so calculating the change in allele frequency from generation T to generation T plus 1, given S and given the allele frequency of at generation t. Okay, as a function of the allele frequency and the selection coefficient, calculate. Our goal is to calculate change in allele frequencies, come up with a general formula. And how can we do that? We can use the description of Hardy-Weinberg allele frequencies, but adding on top of that selection. Okay, so if there was no selection, the genotype frequencies would represent the would be represented by the Hardy-Weinberg frequencies. But then once selection happens here, right, some individuals of certain type die more frequently, that changes the frequency of the genotype, of one genotype by 1 minus S here, right? So the frequency of A2 homozygous individuals decreased by 1 minus S. If the beneficial allele had not been dominant, here the beneficial allele is dominant, and that's why heterozygotes have the same fitness as homozygous beneficial individuals, and their fitness are both 1. If that had not been the case, but the beneficial allele had been recessive, then the heterozygotes, we would just change this formula by adding 1 minus S and by multiplying the heterozygous frequency also with 1 minus S. But here it's dominant, so we just multiply the frequency of homozygous recessive individuals. Okay, and now these individuals whose frequency has been altered by selection, these individuals will be creating next generation, right? And therefore, P prime, the next generation allele frequency of A1, P prime will be somewhat different from P of the previous generation. So our target is to describe a formula for delta P, so change in allele frequency as a function of P and S. And this is not too complicated. In fact, so we just first have to think of the 3 genotypes and their frequencies. So frequency of the A1 genotype is P squared. Nobody has died here, but its relative frequency has increased because the total number of individuals in the population is now P squared 2P cubed plus Q squared times 1 minus S, right? Because these guys have decreased. So this is the frequency of the A1 individuals. This is the frequency of the A1A2 individuals. And the frequency of A2 individuals, it will be Q squared 1 minus S, and also again divided by different number here. And sorry, different number meaning it's the same denominator, of course, for all of them. But it's different from the initial frequencies. Okay, and let's say the altered post-selection frequencies. And so these individuals will be carrying A1 in 2 copies. These individuals will be carrying A1 in 1 copy. These not. And given these numbers, we can calculate the A1 frequency of the next generation, right? This plus half of this will be the A1 frequency in the next generation, P prime. Okay, so this part, the denominator, actually, it boils down to this more simple version, 1 minus Q squared S. How this is derived, you should be able to figure out. It's not complicated. What you just need to have to keep in mind is the sum of P squared plus 2P cubed plus Q squared is 1, right? Will be 1 by definition. And given that, you should be able to derive, you should be able to reorganize this part of the equation into, simplified to 1 minus Q squared S. This is the same thing. Yeah, the same values, just simplified. And oh, sorry. just a second. Here we are missing the minus sq squared part. Yeah, here it just says q squared. This is before selection, of course. So I think it could be a mistake of the book. Sorry. Let me just add it here. Minus s times q squared should be, yeah. Okay, and now let's calculate p prime. P prime will be, again, the frequency of A1 heterozygotes plus half the frequency of these guys, and p squared plus half of 2pq, because these guys carry the A1 allele. They carry only one copy of the A1 allele. And again, divided by the same denominator, we get p divided by 1 minus q squared s. How this can be simplified to just p, again, you should be able to derive. And if you cannot, you can, of course, ask, but please, yes, think about it for a couple of minutes, and it should be straightforward. You can also discuss with your friends, of course. Okay, so p prime is p divided by 1 minus q squared s. So this value will, of course, be, let's look at it. If s is a positive value, this will be overall larger than than p itself, right? The result of this division will have a higher value, because here the denominator will be smaller than 1. If the denominator is smaller than 1, then p prime will have a value greater than p, obviously. And then if and also keep in mind that the q is always something between 0 and 1. And then we can also calculate delta p. Delta p is the difference between p prime and p. So the difference between p at the next generation minus p in the previous generation. That difference is simply boils down to this value. How do we calculate that? We say we just subtract from this value p, and then you just reorganize and you end up with this value. Again, this is exercise for you. You can do it yourselves. And okay, so let's study this formula. So it depends on p, as you see, and q is just 1 minus p. So it depends on the allele frequencies. Q is not something independent. It depends on p, and it depends on s. And the higher the s, the larger the value here will be, and higher the s, the denominator will also be smaller. So the stronger the selection coefficient, the change in allele frequency from one generation to another should be higher, right? That is obvious. How the relationship between delta p and p itself will be, that's somewhat less straightforward, because there's both p and q here. Yeah, it's a bit more difficult to guess. I mean, not so intuitive. The relationship with s is obvious, but not with p. But we'll see that relationship soon. Okay, so this formula of delta p describes change via selection in one generation, just one generation. You see that higher s, faster change will be, the difference will be stronger. As q decreases, you might guess that the change might slow down. So this part will be smaller. So denominator, as q decreases, denominator will grow. It will approach 1. And here it will also, if P increases, the decrease in Q will be more strong. So change we can expect as Q approaches 0, the difference will become smaller and smaller. And actually, you can see if you plot, you could also do this in the R environment. You can plot change in P and update P each time and see how this this will come out in the long term across generations. So here we see such simple simulation done. P calculated across generations, starting from 1%, I think, starting from 1% at different selection coefficients. Here we see selection coefficient 10%, what we had assumed might apply to our mouse case. And as you see, so it changes first somewhat slower, and then it becomes fast, and then it slows down again towards the end as Q, the alternative allele, is becoming rarer and rarer. As it approaches 0, the change also approaches 0. Yeah, so the time for fixation will be pretty late, but the beneficial allele will become widespread, very widespread in already 300 generations, as you see. If selection coefficient was stronger, like 0.7 here, then change, the shape is kind of similar, but it is just so much faster. If it were somewhat more intermediate, so it looks like this. Yeah, so depending on selection coefficient, we have a different rate of responses as we predicted. And the final fixation, though, it takes time because as again, Q approaches 0, the change here will also be smaller. And this is a deterministic model, so it doesn't include drift, genetic drift. Okay, so this was an example of directional selection, a directional selection model when the beneficial allele was dominant. Okay, so which is described by this plot here. So we see fitness on the y-axis, fitness of the 3 genotypes, and the heterozygous ones also have fitness 1. Homozygous homozygotes of lower fitness are recessive type. But we can have 2 alternative situations. We can have the recessive, the low fitness allele being dominant and the beneficial allele being recessive. Okay, then the heterozygous will have also lower fitness. We can also have the co-dominant situation, or actually, as you see here in third-year genetics, we learned that co-dominant is the situation where the heterozygous express both alleles, and intermediate dominance is the situation where heterozygotes express the phenotype in an alternative, in intermediate stage. So intermediate dominance would be a more correct term to use here. So if heterozygotes have intermediate fitness, then yeah, you can have potentially different response. So how will these different scenarios affect this pattern, or will they affect this pattern? And think about this for a second. And answer is yes, they will affect quite dramatically. So here you see again, directional selection for a beneficial allele, totally deterministic. The only difference here between these 3 curves is selection coefficient is also the same on the beneficial allele. The only difference is one of the curves describes beneficial allele, the beneficial allele being dominant. The other one describes another one describes beneficial allele being intermediate dominant, and the third one describes situation where beneficial allele is recessive. And which one is the recessive case? Can try to imagine, can think of it for a second. It's as follows. So the red one represents the dominant situation, what we saw. And when the allele is dominant, it takes long time for it to fix. And that is simply because the heterozygotes have the same fitness as homozygous beneficial allele genotype individuals, right? So the A2 allele is basically hidden behind the A1 allele in heterozygotes, so they're not exposed to selection very efficiently. Just the opposite happens in the case where the beneficial allele is recessive. When the beneficial allele is recessive, when the allele is rare, most of the alleles will be in heterozygotes, right? Most of the beneficial alleles will be carried in heterozygotes. So only when two heterozygotes mate and produce A1 individuals will the frequency of A1 individuals increase. Otherwise, A1, the beneficial effect of A1 will be hidden behind the low fitness effect of A2. And so it will take long time for this for the beneficial allele to show its effect and increase in frequency. But once it starts spreading, it will increase very fast, and then it will fix very fast because the A2 allele cannot hide behind these ones have much higher fitness. And in fact, the most efficient selection happens when the beneficial allele is codominant, because it will start increasing really fast, but then it will also fix relatively fast. And that is because the A1 allele, the copy number of the A1 allele is directly influencing fitness. At the start, when A1 is at low frequency, it will be in heterozygotes, but it will still increase fitness. So the heterozygotes fitness will, the heterozygotes will spread, and A1 will spread. And after some point, there will be more heterozygous matings and more homozygous A1 carriers, more homozygous A1 individuals being produced with higher fitness, and then A1 will continue to spread. So we will have relatively efficient selection, as you see here. Yeah, so why we have most efficient selection in the codominant case? Because there's none of the alleles, the bad allele or the good allele, they cannot hide behind each other, as happens in the dominant or recessive case. The selection can directly see the phenotype. Okay, here we have, this is not from the book, but for your information, which is kind of intuitive. So we can also write a more general formula for the for directional selection. Instead of A1, A2, here we have the allele, two alternative alleles are shown with the capital A and small case a. So these are heterozygous, homozygous again. And W is the fitness. Okay, W is a common way to describe fitness. Okay, yeah, it's good to know. And here this formula describes the frequency of the parents after selection happens. So fitnesses will be determining what how much selection is happening. So this, in the previous case, for instance, the fitness of the homozygous recessive individuals would be 1 minus S, and the fitness of the heterozygous and the homozygous dominants would be 1, right? So this would burn down to what we just saw. And then here on top, we would again have fitness 1, fitness 1. And this will be showing, so this part of the equation shows the frequency of the big A allele, like the A1 allele in the previous case, the capital L A allele in the next generation, minus P, the frequency of the capital A allele in the previous generation. The difference is delta P, again, change in frequency. It's the same thing, but this is more general. So here you can also, with this formula, you can also describe the change in allele frequency when the fitness of heterozygotes, for instance, is the fitness of heterozygotes is the same as fitness of homozygous deleterious alleles, allele individuals. So you could replace this with 1 minus S or anything. Yeah. And this formula can be rewritten, reorganized in this manner. Here, W bar indicates average fitness. So the fitness of the population, just this denominator, it's a shortcut for the denominator. The numerator here has been reorganized, and we have this type of formula, and you should be able to see like how this formula is actually equal to this formula for the case of the dominant allele, when W, the fitness of capital A individuals, and the fitness of heterozygous is 1, and the fitness of homozygous deleterious allele individual is 1 minus S. You should be easily deriving how this numerator turns down to this this numerator, the formula we just studied. Okay. So here in this directional selection model, we can also ask what at what point the population will reach equilibrium. Equilibrium, as you remember, is the stage where there's no change anymore any further. And here we have a formula describing change from one generation to another. So the equilibrium state will be reached when that change is 0. That is, delta P is 0. When will delta P become 0? It could become 0 when S is 0, right? But S is, we already assume, fixed in this case, and a positive number. So this can become 0 only when Q is 0, right? When Q reaches 0, the state will reach, the population will reach equilibrium, and it will be a stable equilibrium. Once it reaches there, there's no polymorphism rate left. A1 or capital A has fixed, the alternative has been lost. And so, yeah, the population will remain there until new mutation, whatever happens. Okay, so the equilibrium state under directional selection is loss of one of the alleles, we said. Can you also imagine a type of selection where the equilibrium state is polymorphism, where both alleles remain? Could we have another type of selection where both alleles remain in the population? And indeed, we discussed this. So Bayesian selection is the name, is the umbrella name of different selection regimes which lead to both alleles remaining in the population. And one specific case is of that is heterozygote advantage or ovodominance. They refer to the same thing, where the heterozygotes have higher fitness compared to the homozygous, and that is regime which leads to both alleles remaining in the population. And this graph, it's similar to the graphs we saw here, describing allele frequency change over time. Is a bit weird, I'm sorry. It describes the results of 2 experiments in a single graph. Okay, so one experiment is here. We start, we're looking at the frequency of the A1 allele. As you see, the heterozygotes have higher fitness A1 has lowest fitness, A2 has somewhat higher, but still lower than heterozygous. In this condition, if we start with a population which has low, let's say 1% A1 frequency, everybody is A2, there's just few heterozygotes which carry A1. How will the frequency of A1 change? Well, it will increase because, as you see, the heterozygotes have higher frequency than A2 individuals, which constitute most of the population. So it will increase, but then eventually it will reach an equilibrium state, and it won't go any further because A1 individuals who might be produced from the mating of two heterozygotes, they have lower fitness, right? So there will be an upper limit to how many heterozygotes we have in the population. The population will reach a situation where there will be heterozygotes and homozygotes of both type, and the fitness differences will create this kind of equilibrium state. You can also start the experiment from the other side. You can start the experiment with most individuals, most of the gene pool having A1 at high frequency, and only small frequency of A2s, and then the A1 frequency will decrease because, again, heterozygotes have higher fitness, will decrease, and then it will reach a state where again, the frequency of the three genotypes are in balance given their fitness differences. What this balance is, we'll learn in a second. Just briefly, you already know this, that we have good examples of overdominance heterozygote advantage. Sickle cell anemia was initially proposed to be such an example, and it was noted that it occurs in regions where there's high malaria frequency historically, and it was proposed to be selection response in human populations that could have potentially protected against malaria, and it later turned out to be the case. Indeed, in areas of malaria, individuals who are heterozygous have higher fitness. Their red blood, they don't show the sickle cell phenotype. The red blood cells are more or less normal, but still slightly abnormal. That makes malaria grow inside them. I forgot the name of the parasite. Anyway, Plasmodium. Plasmodium grow inside their red blood cells. Plasmodia growth inside the red blood cells, inside heterozygote red blood cells, is more difficult, and that protects them against malaria, but they don't show the sickle cell phenotype that gives them relatively high fitness. Yeah. So overdominance is one cause of one path to having balanced polymorphism. So polymorphism remain in the population by selection. Selection driven by heterogeneous environments where one allele is advantageous in one environment, another allele in the alternative environment. So environmental heterogeneity or temporal heterogeneity, let's say seasonal cycles where one allele is beneficial in the cold and the other allele beneficial in warm environments. These kind of situations can also lead to long-term maintenance of polymorphism in the population. Underdominance, we'll see in a second, is yet another, it can contribute to polymorphism in the population, but it's an unstable equilibrium, so it will be short-lived. And another important contributor, important process, is negative frequency dependent selection. We'll talk about this in a second after a few slides. Okay, so how can we guess this? How can we model this allele frequency change over time as a function of the fitnesses? In fact, we can simply use the same formula I was showing you. This is from a population genetics book, and if you're interested, I can also share with you the link. It can be found as PDF. So given this formula, this is a simplification of um, uh, the delta P formula we said, which describes how P changes, the frequency of the P capital A allele changes as a function of both its frequency and the difference of fitnesses. So here, if we just plug in the fitnesses of the three types, of the three genotypes, this will also help us calculate the change in allele frequency under Balancing selection, just like in the direction selection. It's the same thing. Okay, and then given this formula, so if we have a formula for Balancing selection, can we estimate the stable state? Can we estimate the equilibrium under overdominance? So under overdominance, the equilibrium under overdominance should have three conditions. So as any equilibrium, it should have delta P 0. So change in allele frequency should be 0, right? As is implied here. So you see increase and then there's no change. Decrease and there's no change, right? And if you would push the system in one direction, so if by migration, for instance, there were more A1 alleles introduced in the population, if this moved upward, it would go back down, okay? Or if it were moved downward by migration or drift, it would move back to this point. So selection would maintain the same equilibrium value, okay? This is the equilibrium value. And now we want to calculate, so I suggested that this formula is sufficient to calculate the delta P and the Balancing selection as well. So if that's the case, can we estimate that allele frequency at which we see equilibrium, at which there's no further change? So no further change, that would require delta P to be 0. But also, of course, delta P could be 0 by having P or Q 0, obviously in this formula, right? But that's not something we are interested in. We want to have equilibrium where both P and Q are non-zero. P and Q should be non-zero and delta P should be 0. How can we derive that from this formula? Well, what can we do? So this part is non-zero, obviously. How about this part? So this part, if we can rearrange this part to be 0, then we have found that, or I should say, given the selection coefficient and the fitnesses of the three genotypes, if we can find the P and Q, well, P and Q are basically just inverses of each other, right? If we can find the P value, the allele frequency at which this part of the equation is 0, then we have our equilibrium frequency value. So basically, just a repetition of what I said. So this part should be 0 on top, and we can, of course, rewrite this by substituting 1 minus P instead of Q, right? Same thing. And then here, instead of big A and small a, just following the book, I've used 1 and 2 for the fitnesses. W11 means the fitness of A1, okay? And we then just need to reorganize. We can open up this multiplication, and then, so to do the multiplication, we have this equation then, and then we can reorganize by how we can collect all the ones which, all the items which contain P under this P parentheses, So we have something like this here. And then on the other hand, we also have this part. And then what we just need to do is, move P to one side. So move this this part over beyond, and then move this part below and multiply by -1 both sides, and we get this result. So the frequency of P under this condition, so to achieve these conditions, we would need to have P as a function of the allele frequencies, P as the difference between the fitnesses of the heterozygote and the homozygote, the alternative allele, the second allele, and some function of the fitnesses again in the denominator. And so this seems to give us the allele frequency at that would be the stable equilibrium. For instance, in this case, we had fitness of the 1,1 type 0.8, heterozygote 1, fitness of the 2,2 type 0.9. What would be the equilibrium allele frequency for the A1 allele? Okay, this would be 1 minus 0.9, right? And then here we would have, again, 2 minus 0.8 minus 0.9, and we would have something like 0.03 here, right? 0.3, and here 0.1. So what's that? 1/3, basically. 1/3. Yeah, this is the expected equilibrium value. Okay. And if you can also notice if in this model, if the two genotypes had equal fitness, so two homozygous genotypes had equal fitness, then what would be the equilibrium? If the two homozygous genotypes had equal fitness, what would be the equilibrium allele frequency? What would be the equilibrium P? Okay, this you should be able to solve, and if not, we can discuss during presentation. Okay, so one point which is important here to mark again, if once the system reaches the state, if it remains undisturbed, it remains the same till infinity. If it is disturbed meaning the frequency of one of the alleles increases for whatever reason, selection, migration, whatever. Sorry, not selection. We're assuming there's only Benson selection going on here. Could be by migration, could be by drift, for instance. Selection would push it back to the same level as long as the same fitness coefficients remain. And then yeah, so this is a fully stable equilibrium, okay, with both both alleles remaining in the population, but it leads to genetic load in the population. So it's not a very nice type of equilibrium for the population in the long term. For a population that is, for instance, that has achieved higher fitness by introducing by a mutation which has overdominance, where heterozygotes have higher fitness, versus a population which has achieved the same adaptation but without overdominance, where just one allele in homozygous type creates the same effect. Let's say both is healthy and protects against malaria. This population will be the one without Benson selection, the one without this balanced equilibrium will be more successful. Why? Because as you see, so in this situation, so heterozygotes, there will be a high number of heterozygotes, but also both types of homozygotes will remain in the population, right? And these will have lower fitness. And so the population will be a mixture between high fitness and low fitness individuals. And so this makes the population under balancing selection less successful in the long term. So perhaps for that reason, we don't, although we do find examples of overdominance in many different organisms, it's not, it doesn't seem to be the most common way for adaptation. If directional selection, if an allele arises by mutation that has higher fitness compared to all these three, or doesn't even have to be a higher fitness. If we have a 3 allele which has the same fitness as the heterozygous, but in homozygous states, this will be selected. Okay, it will outcompete the A1 and A2 alleles. Okay. Yeah, there's also alternative derivation in the book for the same formula, how the equilibrium allele frequency is calculated in the Benson selection situation, but I think it's less intuitive than the one I was trying to show you. So, but yeah, you can also try to follow their logic. Okay, underdominance is another type of relationship. Situation where it's basically the reverse of overdominance, where the heterozygotes have low fitness compared to either homozygote. And again, it has a value which corresponds to an equilibrium state. So at a certain p, and it's basically the same p, given the allele, given the fitnesses, you can calculate the equilibrium allele frequency at which the population will be in balance, but it's an unstable balance. And if the system is pushed in one way, if one of the alleles is at higher frequency, it will move in that direction. If the system is just pushed slightly in the other direction, if the other allele becomes more frequent, it will move in the other direction, and the end result will be loss of one of the alleles and fixation of the alternative allele. It's an unstable equilibrium in that sense. Okay, it's not. It doesn't bounce back. It doesn't remain to the equilibrium state. And why that is the case, you should again be able to see. Again, reading the book can also help. These examples of underdominance we can see in when lineages, populations, or species, populations which are somewhat distinct or closely related species, when they mate, so individuals of either type are well fit, are adapted, but the hybrids sometimes can have lower fitness. An example of some butterfly. So they are members of the same species, but different forms. And this is a case of mimicry, where the one type resembles one type of local toxic species, so it can avoid predators. The other type resembles another toxic species, but when they mate, the hybrids don't look like either, so they have lower fitness. So there will be, in this case, selection against hybridization, strong selection against hybridization. We'll also discuss that in 2-3 chapters ahead in the speciation. So this creates, as we mentioned, the unstable equilibrium, and in the long term, one of the alleles should be lost, the other one survive. Okay, here we stop.